Welcome dear students. Today we are going to discuss the nucleic acid chapter. So what is the nucleic acid? Let's discuss the brief introductory part of the nucleic acid. Discovery. The nucleic acid has been discovered by the Friedrich Mischer. What are the type of nucleic acid? That is the DNA and an RNA. What is the building block? That is the nucleotide, and the nucleotide has been divided into component. What are the two components? That is the nitrogenous bases. What are the nitrogenous bases? Let's see what the nitrogenous bases is. It is the purine and the pyrimidine. The purine contains the two parts, the adenine and the guanine. And why the pyrimidine contains the cytosine, thymidine and uracil. The second is the phosphate group, and the third is the sugar. And the sugar is mainly a pentose type. And structure and conformation is at envelope or twisted. What is the bond present in the nucleic acid? That is the phosphodiester bond and the glycosidic bond. The glycosidic bond is of two types, the anti and the zinc. The minor bases. The minor bases of the DNA and the tRNA. We discussed this in subsequent class. The properties of the bases. The nucleic acid is history, structure of double-stranded DNA, interaction stabilizing the 3D structure of the DNA, unusual structure of the DNA. The form of the DNA, that is the A form, Z form, B form. The nucleic acid chemistry. In the nucleic acid chemistry, we are going to learn what is the viscosity, denaturation, renaturation, hyperchromic, hypochromic effect, melting temperature, and the factor affecting the Tm. T is the melting temperature. What is the GC content? It is the guanine and the cytosine content that is present in the DNA structure. The ionic strength and the pH. What are the properties of the nucleotide bases? The first is the weakly basic. The second one is the aromatic molecule. The third one is purine planar, and the pyrimidine is nearly planar. The third one is tautomerization. The fifth one is UV absorption. Sixth is hydrophobic at neutral pH. And the seventh is hydrogen. It is a hydrogen. Sorry for the wrong word. It is a hydrogen bonding pattern in base pair. Okay. The unusual structure of the DNA. It has a 5 type of structure mainly that is present in the DNA. That is the bends, palindrome, mirror repeat, hairpin cruciform, triplex DNA, and the G quadruplex or tetraplex. The interaction stabilizing the 3D structure of the DNA. Hydrophobic stacking interaction, van der Waals interaction and dipole-dipole interaction, hydrogen bonding between the complementary bases. And the fourth is the electrostatic interaction. Then we see some basic of the nucleic acid. As I told you in the beginning, it has been discovered by the Friedrich Mischer. And he discarded from the surgical bland from the nuclei from the pus cell. From where he discovered the nucleic acid? From the pus cell, which is the leukocyte. And the leukocyte are the WBCs, as we know that. He called it as a nuclein. What is the nuclein? The nuclein is just a combination of a protein plus phosphorus containing the organic acid, and which is called as a nucleic acid. The nucleic acid are of two types, DNA and RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. What is the nucleotide? Nucleotide are the building block of nucleic acid. Nucleotide is also known as the polynucleotide because why it is so called the polynucleotide? Because many nucleotides are joined together to form a polynucleotide structure. The nucleotide 3 component. The first is the nitrogenous base. The second is the pentose sugar, and the third is the phosphate. Okay, let's see what is the nitrogenous base, what is the feature and what are its type. The feature is they are heterocyclic, planar, relatively water insoluble. Remember this point. And aromatic. What are the type? The type is purine and pyrimidine. Over here it is given the structure of purine and pyrimidine. When we do the numbering of the purine, always numbering is done in an anti-clockwise direction. You can see. While in the pyrimidine, there's always the numbering is done from N1 into the clockwise direction. While in the purine, it is an anti-clockwise direction. Okay. This is a 5 structure of adenine, guanine, cytosine, thymine and uracil. When we see the adenine, it is also known as the 6-amino purine. Why it is so called the 6-amino purine? Because at the 6th position, carbon number 6, there is an amino group present. Therefore, it is known as the 6-amino purine. In the guanine structure, at the 6th position, oxygen is present, and at the 2nd position, the amino group is present. Therefore, its name is 6-oxy-2-amino purine. Okay. In the cytosine structure, at the 6th position, oxygen is present, and at the 4th position, amino is present. Therefore, it is known as 2-oxy-4-amino pyrimidine. It is the cytosine. In the thymine, at 5th position, methyl group is present, while in the 2 and 4th position, oxygen is present. Therefore, it's known as 5-methyl-2,4-dioxypyrimidine. And the structure of the uracil is like this. In the uracil, at 2nd and 4th position, oxygen is present. Therefore, it's known as 2,4-dioxypyrimidine. Always remember, the name purine is very small, but its structure is a big. While the pyrimidine, its name is a big, but its structure is very simple and is small. Okay. What is the pentose sugar? The pentose sugar is of 2 types. 2'-deoxy-D-ribose. While as the RNA is D-ribose sugar. Why it is so called? What is the D? D is a dextro. Okay. Dextrorotatory. Its rotation is dextrorotatory. Okay. In nucleotide, both type of pentose are in the beta furanose. What is the beta furanose? Beta furanose means it is an enclosed 5-member ring. Okay. What is this difference between beta D-ribose and beta D2-deoxyribose? At position number 6, you can see there is a hydroxyl group is present. Okay, therefore it is known as a beta D-ribose. While in the deoxyribose sugar, at position number 2, there is no hydroxyl group, only hydrogen is present. Therefore, its name is beta D2-deoxyribose. Okay. Pentose ring is not planar but puckered in 2 bases conformation. What are the 2 conformation that is found is? It is called as envelope and the second is twisted. What is the envelope? In the envelope form, 4 carbon are coplanar. Coplanar means all the 4 carbon are present in the same plane and the 5th is away from the plane. Twisted. In twisted form, the 3 carbon are coplanar. 3 carbons are in same plane and the other 2 lie away from on the opposite side. The twisting is C2' or C3' related to the other carbon, resulting in the twisting form of the sugar. You can see the envelope form is 3' endo and 2' endo. 1, 2, 3, 4. You can see 1, 2, 3, 4, 5, right? The 3rd carbon is out of the plane, therefore it is known as the envelope form of C3 endo. In the second diagram, you can see the envelope form is C2 endo because the C2' is out of the plane. In twisted form, one is the 2' carbon and the C3 carbon is out of the plane. Therefore, the C3 is endo, but the C2 is exo. Okay. In the twisted form, C2' endo and C3 exo, because the C2 endo over here and the C3 exo. Endo means from inside, interior, and the C exo mean outside the plane, right? They are in opposite direction. Therefore, to discriminate between them, we name it like an endo or exo. Okay. Okay. What are the nucleosides? The sugar and the base join to form the nucleoside. The base is joined covalently at the N1 position of the pyrimidine and N9 of the purine in the N-beta galactosidic bond to C1 of the pentose, and bond formation takes place by the removal of water. Always remember, when there is a bond formation takes place in the beta galactosidic, there always a removal of water. The nucleotide, the phosphate are esterified at the 5' carbon of the nucleoside. The nucleotide can have 1, 2 or 3 phosphate group, designated as an alpha, beta, gamma for the first, second and third respectively. Okay. In the diagram given below, you can see, okay, In this diagram given below, you can see at 5th position, the phosphate group is attached. There are three multiple. I'm just shown you a single phosphate group, but there is a series of 3 phosphate groups, and they are named according to alpha, beta, and gamma, right? Due to the rotation of the glycosidic bond. What is the glycosidic bond? This is the glycosidic bond that is formed here. Okay, when this glycosidic form, there are 2 conformation possible. Sync when it is on the same side, and there's anti when there is an opposite side. This is the diagram of adenosine. When the base is on the same side like a sugar, it is known as a sync adenosine. When it is opposite direction, this is known as the anti. Okay. The nucleotides are found in the monomeric unit of the DNA and RNA. However, other functions include the formation of energy currency like ATP and GTP. It acts as a precursor for several important coenzymes like NAD+, NADP+, FAD, and a coenzyme A. It also serves as a precursor for the secondary messenger like cyclic AMP, CGMP. Okay, what is the secondary messenger? Secondary messenger are the messenger that transmit they are present in the cytoplasm and they transmit the signal to the nucleus so that they can synthesize the functional protein for the cell to function, to act correctly, right? In the pentose that define the identity of the nucleic acid, if the nucleic acid contains 2'-deoxo-D-ribose, it is a DNA by definition, even though it contains the uracil. While in your book, you can see the CMP, UMP, TMP, AMP, and GMP. What are these? They are the monophosphate, uridine monophosphate, thymidine monophosphate, adenosine monophosphate, and guanosine monophosphate. Okay. Some minor purine and pyrimidine bases. These are the minor purine and pyrimidine bases. You can learn it. They are the pseudouridine, dihydrouridine, ribothymidine, 4-thiouridine, 3-methylcytidine, and N4-acetylcytidine. Okay. There are some purine derivatives: 1-methyladenosine, ionosine, N7-methylguanosine, and N2-N2-dimethylguanosine. Okay. The phosphodiester bond. The 5' phosphate group of one nucleotide join unit is joined to the 3' hydroxyl group of the next nucleotide, creating a phosphodiester linkage with the elimination of water. When the phosphodiester bond form, when the 2 nucleotides are added, the 5th position and the 3rd position, always remember that, with the elimination of water. This is a phosphodiester linkage. Okay, this is a 1, 2, 3, 3rd position, and this is the 5th position. The alternate phosphate and pentose hydrophilic side group. What are the backbone that are present? This is a phosphate, this is a phosphate, and the pentose hydrophilic side chain groups, bases. Okay. The important things to remember: the properties of the nucleotide base affect the 3D structure of the nucleic acid. Okay. How this is possible, I'm going to teach you this in next lecture. Okay. Free pyrimidine and the purine are weakly basic because of the N atom. That is why it is called as a base. These bases are aromatic molecule, property with important consequences for the structure, electron distribution, and light absorption of the nucleic acid. What are the light absorbing group that are present in the nucleic acid? That are what? Do you have any idea, students? What are the light absorbing functional group that are present in the nucleic acid structure? Okay. Wait. Pause this video and tell me in the comment box. Do you have any idea? Okay. Its name is tryptophan, tyrosine, and phenylalanine. Always remember this. When I am going to teach you the amino acid chapter, I will explain to you in great detail what are they and how they work and how they are important, telling the property of the nucleic acid, or what is the structure of the protein when they are present in nucleic. Okay. The electron delocalization among the atom in the ring give most of the bond partial double bond character. Thus, purine is a planar with a slight puckered. How it is puckered, I have told you. Okay. Free bases may exist in 2 or more tautomeric form depending on the pH. The lactam form predominant at pH 7. Other form become predominant as pH decreases. All bases absorb UV light, and the DNA are characterized by the strong absorption of the wavelength at 260 nanometers. The purine and the pyrimidine bases are hydrophobic and relatively insoluble in water at the near neutral pH of the cell. At acidic or alkaline pH, the bases become charged and the solubility in the water increases. Okay, thank you so much.